Hi, I'm Jan Witkowski from Cold Spring Harbour, and I welcome you to what is the 86th Cold Spring Harbour Symposium on Quantitative Biology. I always have to think hard to get those right. And after a gap of 2 years, it's wonderful to have people back here again in person. The topic of this year's symposium is genome stability and integrity. And I'm delighted to have Andrew Jackson here from the University of Edinburgh, who is an authority, of course, on such a topic. Andrew, one of the things, reading your abstract, because of course you've not given your talk yet, I had never heard of embedded ribonucleotides in DNA.
In DNA, yes. Well, not many people have, and I mean, even though it's now 10 years since we published the Cell paper describing them in mammals, and even longer that the likes of Tom Kunkel identified them in yeast, there's still not a great familiarity with them in the textbooks or even among many scientists studying genome integrity. But yes, it's surprising that newly replicated DNA has a huge number of ribonucleotides within the DNA, over 1 million per replicating cell, and that makes them the most common endogenous lesion within the genome. You know, they get misincorporated by the replicative polymerases, which have very high affinity for deoxynucleotide triphosphates, but because of the vast excess of nucleotide triphosphates, they occasionally incorporate maybe one in every 1,000 base pairs a ribonucleotide.
So to sort of simply, there is just so many more ribonucleotides that they not outcompete. That's not the right word, but outplay or something. So these are fairly common. So how, if I may put it this way, how come nobody had found them until you did? What was the sequence of that?
So, I mean, again, a lot of this has its origins in yeast genetics, and our role has been to translate that into a mammalian setting. For us, it came about from studying human genetic disease, rare disease, Mendelian disorders. I'm a clinician and clinical geneticist, and one of the disorders I work on is Aicardi-Goutieres syndrome, which is a rare inflammatory disorder in early childhood, which causes brain inflammation, which looks very like congenital viral infections. And myself and my good friend and colleague Yannick Crow found 4 genes for this condition back in 2006, 3 of which encode ribonuclease H2. And this enzyme is a key component of a pathway which removes ribonucleotides which have got into the genome, such that you don't normally see them. But in the absence of this enzyme, you suddenly discover there's a huge amount of them present. And this pathway has been, which removes ribonucleotides, is called ribonucleotide excision repair. And essentially sits alongside those pathways which we know about, which excise abnormal bases, so base excision repair, nucleotide excision repair, and mismatch repair, to recognise these ribonucleotides and remove those. So it's really recognising an abnormal sugar in the sugar backbone of DNA.
When you said there were 1 million ribonucleotides per replicating cell, is that the sort of the number of them that you would find in a normal cell, as opposed to the mutated cells in this condition?
So you'll see they'll be there in the normal cell, but you won't be able to detect them because of the efficiency of this enzymatic process of removing them. So yes, so they're there, but transiently. So they're rapidly removed. But if you take away the enzyme, which is the first part of the pathway to remove them, then you see them. You see all these ribonucleotides within the genome.
So in the presence of this enzyme in normal cells, what are the consequences of having all these ribonucleotides scattered around?
So, for a long time we wondered whether there was any consequences at all, but our most recent work has suggested that there are consequences. And these come about because another enzyme in the cell, the topoisomerase 1 enzyme, can actually also cleave at ribonucleotides embedded in the genome. But instead of initiating an event which cleanly removes and replaces it with a deoxynucleotide, topoisomerase 1 results in a dirty end single-strand nick, which is hard to repair by the cellular machinery. And then, through additional steps, that can end up causing mutation through additional cleavage by the topoisomerase 1 enzyme, resulting in small deletions. And our most recent work has shown that this results in a recognizable deletional signature in cancers, both cancers which are deficient in the RNase H2 enzyme, but also in a broader range of cancers where RNase H2 is present. And surprisingly, we also find that these deletions occur in normal cells, and from looking in the human germline by looking for de novo mutations, we can pick up this signature as occurring in early development in humans.
What's the characteristic of this signature?
It's a short deletion signature, so 2 to 5 base pairs in size, and it also occurs at certain sequence contexts where there's a repetition. So, for instance, you had 2 nucleotides which are repeated once. So, for instance, you might have a TC sequence which is repeated twice, so TC, and one of those TC dinucleotides is where the deletion is occurring. And this comes back to the mechanism by which topoisomerase 1 cleaves, and then its second cleavage event, which causes a deletion, will result in a deletion only if it can realign the sequence, a sequence realignment process, which means that if you've got a repetitive sequence, it can do that much more efficiently. But it's not like the repeats you commonly think of, short tandem repeat microsatellites, which are much longer and associated with mismatch repair, induced instability. So it's a distinct signature. The other things we know about it is it has a certain nucleotide composition, a TNT motif. And
a TNT motif?
Yes. So thymidine, any nucleotide, thymidine. And that comes about because of topoisomerase 1's preference for cleaving at a thymidine residue. And the other thing which is quite surprising is it's not uniform across the genome. It occurs at sites of high transcription. So we can see a correlation with transcriptional, highly transcribed genes. And so, in effect, what we're seeing is a process which has been seen previously in yeast and bacteria called transcription-associated mutagenesis, and showing that it actually is occurring also in mammalian cells. And certainly my prior sort of assumptions had always been that transcribed genes were protected by, transcription-coupled nucleotide excision repair. So it came as a surprise to me that actually there are actually reasons why such sequences have increased mutation rates as well.
Yeah, I was taken by your use of describing topoisomerase 1 as an endogenous mutagen. I don't think of cells having endogenous mutagens.
Maybe it's a slightly provocative terminology which I'll get taken up on. But yes, I mean, I guess a lot of work in genome stability has been thinking about exogenous agents which causes mutations, but I think, and we're going to hear other really nice talks, for instance, from KJ Patel about endogenous aldehydes as a source of mutation and genome instability in the genome. And I guess it's of interest to me is what are the things which causing mutations in cells which are not coming from external sources.
But these endogenous, let's call them changes rather than...
Okay.
But presumably not in a regular cell. These endogenous mutant changes are by and large dealt with. Otherwise, we'd all be in real trouble.
Sure, and I think that's something which has taken me a lot of time coming into the mutagenesis field to get my head around, is that actually what you're seeing when you see mutational events is the absolute rare exception to what's normally happening, which is that these processes where we see topoisomerase 1 cleavage at ribonucleotides, most of the time the cell gets it right and repairs it without a mutational event occurring. But occasionally it gets it wrong, and that's when you see mutations. And these mutations are very rare, but we can see the signature of them. And I think it has both a value in terms of, well, mutation drives evolution. Yeah. It also drives cancers. So it's important to understand those processes. But it also gives us a sort of marker of, well, ribonucleotides are occurring physiologically in the genome. These scars are actually giving us a way back to say, well, these processes are happening, very important, and I guess that's another motivation for us to continue to study the processes, because we know they're happening in a physiological in a context.
So presumably these mutations, I'm thinking about their role in cancers.
Yeah.
You detect them in cancer cells, but is that in some way, have they been causative, or is it the nature of the cancer cell that these things occur more frequently in a cancer cell?
I think it's more reflecting the nature of a cancer cell and the processes which are occurring within that cell, rather than that these are necessarily driving the cancer themselves. I mean, mutational events clearly are an important basis for carcinogenesis, but most of the mutations you're seeing, and I guess comes back to this concept of cancer signatures, reflect the processes which have been dysregulated within the cancer cell. And I guess that's another reason for our interest in them, is that as a cancer signature which we've identified, this potentially signifies potential vulnerabilities for the cancer cells which have this signature, the ID4 signature, that may not be present in all cancer cells. Now, one reason for the signature is RNase H2 deficiency, but we don't know what the other predisposing factors are in other cancer types. For the RNase H2 deficient ones, we know that there's a sensitivity to certain drugs, so ATR inhibitors, which are being piloted for as therapeutic approaches, or PARP inhibitors, which are established drugs for targeting HR deficient tumours. So there's potential that you can go from a mutation signature to a targeted therapy. I mean, we don't know yet whether this signature will give those insights in or not, but it's something which we hope our work will lead to in the longer term.
And are you working on any other systems that you're going to mention in the course of your talk on Sunday?
So I haven't yet decided. I mean, I guess I have a wider relevance to this conference than just our work on RNase H2. We also, again, starting from my clinical work, I've had a long-standing interest in microcephalic dwarfism, and a lot of the genes that we've worked on are genes which we'll hear about during the course of the meeting. For instance, replication licensing genes and their association with Mayer-Gorlin syndrome, and genome stability factors as well have been genes which you've identified associated with microcephalic dwarfism. But I think those will have a brief mention, if introductory mention, rather than be a major part of my talk on Sunday.
Andrew, thank you very much.
Thank you.